Hi guys, welcome to DMed lectures. Today we will be discussing about beta lactamase inhibitors, right? Beta lactamase inhibitors. As you all know, the term ACE refers to a particular enzyme, right? So to give you a rough idea about this topic, it means certain molecules which has the capability of inhibiting a particular enzyme known as beta lactamase, right? So now beta lactamase inhibitors inhibit the beta lactamase enzyme, which is actually being produced by certain microorganisms which infect the human body. Before moving on to beta lactamase inhibitors, we should focus on the term beta lactam. What is beta lactam? Beta lactam usually refers to a particular group of antibiotics which are known as beta lactam antibiotics. So if I draw it like this, beta lactam antibiotics, right? So the structural feature of beta lactam antibiotics, the most prominent structural feature will be the beta lactam ring, which is present in its molecular structure. So I'll mark it like this: beta lactam ring. So this beta lactam ring is what gives this particular category its name as beta lactam antibiotics. So this beta lactam ring is what gives these antibiotics its antimicrobial properties, right? So there are certain categories of antibiotics which falls under the name beta lactam antibiotics. So roughly speaking, for an example, penicillins, cephalosporins, monobactams, carbapenems, and carbacifems. So generally speaking, these five groups of antibiotics falls under the category beta lactam antibiotics, which means all of these groups have this beta lactam ring around their molecular structure or in their molecular structure, right? So just remember that it's important. Now, if we discuss about roughly the mechanism of action of these beta lactam antibiotics, what they do is that in the microbe, in the cellular structure of the microbe, we have the cell membrane, right? So just think of this as the cell membrane. There's a particular enzyme or a protein which we call as PBP in abbreviation. PBP stands for penicillin binding protein, right? So this penicillin binding protein or PBP exists in the cellular membrane of the microbe, right? So what these antibiotics do is that they go and they bind to this PBP point, and they usually inhibit this PBP point from its action. So the PBP point, the function of PBP point will be they sort of form the crosslinks in between the peptidoglycan layers, which is important in cell wall synthesis, right? So PBP is involved in cell wall synthesis. So what these antibiotics do is that they inhibit this PBP point from carrying out its action, which means the cellular wall synthesis of the microbe is inhibited, which results in bactericidal activity or destruction of the bacteria. So that is the general mechanism of action of these beta lactam antibiotics, which we talked about earlier. But in some cases, certain microbes which infect the human body. So if I signify a microbe like this, right? Just think of it as a microbe, right? So certain microbes, they have the capability of formatting a particular enzyme known as beta lactamase, right? So this microbe has the capacity of forming this beta lactamase enzyme. So this beta lactamase enzyme, the function of this beta lactamase enzyme, it is favorable for the microbe, not for the antibiotic, right? So beta lactamase enzyme, what it does is that it sort of destroys this beta lactam ring, which is in the molecular structure of the beta lactam antibiotics, through hydrolysis, right? Through hydrolysis, they destroy this beta lactam ring, which sort of prevents this antibiotic from carrying out the antimicrobial function, right? So, so this is not good for the antibiotics because of this beta lactamase enzyme destroys the antimicrobial capabilities. But there are certain antibiotics, for example, if I write it here, like Amphicillin, or for an example, cloxacillin. These kind of antibiotics, which are also beta lactam antibiotics, they have the capacity of being resistant to these beta lactamase enzymes. So especially cloxacillin is usually a penicillin group. It comes under penicillin group, which is known as beta lactamase resistant penicillin. Then cefazolin is actually a first generation cephalosporin. So I'm just talking about examples. So these kind of antibiotics, they have the capability of acting against beta lactamase. They do not get destroyed by beta lactamase, or their beta lactam ring doesn't get destroyed by beta lactamase. So they can carry out their antimicrobial action even though beta lactamase is being produced by the infective agent. But majority of the other beta lactam antibiotics, their beta lactam ring usually gets destroyed by this beta lactamase enzyme. So here's where the inhibitor part comes from, right? So in pharmacology, what we do, as a response to this beta lactamase enzyme, what we can do is that we usually combine this beta lactam antibiotic with beta lactamase inhibitor. So when beta lactam antibiotic is combined with beta lactam inhibitor or beta lactamase inhibitor, the beta lactamase effect on this particular antibiotic is reduced, right? So it is being cancelled by this particular beta lactamase inhibitor section. But don't misunderstand, beta lactamase inhibitor is not an antimicrobial agent. It has very less amount of antimicrobial action. It usually supports this antibiotic to act against this particular microbe which produces beta lactamase, right? So if I give you a quick or a couple of examples about beta lactamase inhibitors, we can talk about, for an example, clavulinic acid, right? Or we can talk about sulbactam, right? Or we can talk about tazobactam. So these type of molecules, these are known as beta lactamase inhibitors, right? So what we do is that we usually combine, like I said before, we usually combine these beta lactamase inhibitors with the beta lactam antibiotic to broaden their spectrum, right? So I will give you some examples about these combination drugs or how they work together. So we have the antibiotic combined with the beta lactamase inhibitor, right? So for example, the first combination drug, so this is the combination drug, right? So because of this beta lactamase inhibitor, the spectrum is broadened of this antibiotic. So for example, amoxicillin. When amoxicillin is combined with clavulinic acid, right? Now this to this combination we have a particular name, we call it augmentin, right? So augmentin, right? So this is actually this is the most classical example for this formula. The antibiotic is the amoxicillin, and the clavulanic acid is the beta-lactamase inhibitor. So the combination of this is known as augmentin. So if I give you another example, ticacillin with clavulanic acid, right? Ticacillin with clavulanic acid, we call it timentin, right? So once again, timentin is the combination drug, so the ticacillin is the beta-lactam antibiotic, clavulanic acid is the beta-lactamase inhibitor, right? So if I give you another example, Amphicillin with sulbactam, right? Amphicillin with sulbactam, the same scenario here, we call it as unacin or unacin, right? So these are some classical examples of combination drugs with the beta-lactam antibiotics and beta-lactam inhibitors, right? So there are lots of other combination drugs out there as well, but I'm just talking about the main combination drugs, which are the classical examples for combination drugs in this topic, right? So I hope you guys understood what I talked about today, and I hope you take out something from it. So if you like this video, just leave a thumbs up and click that subscribe button below, and I will see you again with a new topic and a new video. Thank you very much. Have a nice day.